Hey everyone, let's dive right into this explainer and uncover the incredibly high-stakes biological heist that goes down every single time a virus invades a host cell. We're going to look exactly at how antiviral drugs act as these highly targeted security systems to stop microscopic intruders dead in their tracks. It is an absolutely fascinating pharmacological battlefield. And as you can see here, this perfectly visualizes our central conflict. Notice the syringe representing our clinical intervention going toe-to-toe with an intricate, shielded enemy. That virus, it's a master of infiltration, perfectly designed to slip into our biological secure facilities, our cells, and just take over. Fighting them isn't at all like fighting bacteria. It requires some serious precision engineering at a molecular level. So here is our game plan for today. We'll cover 1. The anatomy of a viral invader. 2. The antiviral drug challenge. 3. Blocking entry and uncoding. 4. Sabotaging nucleic acid synthesis. And finally, 5. Halting assembly and release. All right, let's start with part 1, anatomy of a viral invader. Take a look at the actual blueprints of our cellular intruder here on this basic virus particle, or virion. You've got the outer lipoprotein envelope, you've got the protective protein coat, known as the capsid, and deep inside, the absolutely crucial nucleic acid core. That core right there contains the genetic instructions for the entire hijack. And the antiviral drugs we're going to talk about, they're designed to dismantle or block specific parts of this exact architecture. Once this thing finds a target cell, it doesn't just sit there. It executes this incredibly precise, highly coordinated replicative cycle. It attaches, penetrates, and uncoats its protective shell. Then it actively takes over the cell's machinery to transcribe and translate its own viral proteins, duplicate its genetic material, and assemble a whole army of clones before finally bursting out to infect others. But the good news? Every single phase on this timeline is a potential window for us to deploy an antiviral countermeasure. Moving right along to part 2, the antiviral drug challenge. Now, you might be wondering, why can't we just kill the virus outright? Well, here's the catch. Our antiviral arsenal is virostatic, not virucidal. We can't just poison and kill the virus directly because viruses depend entirely on the host cell's machinery to survive. If we created a drug toxic enough to just destroy the virus, we'd essentially end up destroying our own cells in the process. So instead, we have to limit their growth, effectively starving out the invasion. Because these viruses literally hijack our own cellular machinery, drug developers face this painfully narrow margin of safety. Think about it. The drug has to penetrate the host cell, find the limited viral target sites, which mostly involve nucleic acid replication, and disrupt them without injuring the host. Add in the fact that viruses rapidly mutate to develop drug resistance and can even hide inside our cells in a dormant, latent state where drugs are totally ineffective. You start to see just how massive this pharmacological challenge really is. Okay, part 3, blocking entry and uncoding. Let's look at our first line of defense, basically barricading the doors. Take a look at maraviroc, for instance. It stops HIV by literally blocking the cellular door. It acts as an antagonist to the CCR5 receptor on our cells. So by binding to that receptor, maraviroc prevents the viral GP120 protein from attaching. The virus is completely locked out. We also have gamma globulin, which is given parenterally, providing a whole suite of antibodies to bind specific viral antigens before they even make contact, and arpidol, which steps in to prevent the entry of influenza viruses. Which brings us to part 4, sabotaging nucleic acid synthesis. If the virus actually gets inside, we move to plan B. And this is where we see the absolute genius of a drug called acyclovir, which is vital against herpes viruses. Acyclovir sneaks into the cell masquerading as a normal DNA building block, a guanosine analog. The viral enzyme, thymidine kinase, totally falls for it and mistakenly activates it. Once it's fully converted, it competes to be incorporated into the viral DNA. But here is the brilliant part. Acyclovir is missing a crucial chemical hook, a 3' OH group. So once the virus inserts it into its DNA chain, no more links can be added. Boom, immediate chain termination. The viral DNA polymerase enzyme is permanently jammed. But brilliance in a lab setting doesn't always translate perfectly to the human body. It's actually really important to note that while acyclovir is highly effective against actively replicating herpes viruses, it's pretty poorly absorbed from the gastrointestinal tract. In fact, its oral bioavailability is only about 15 to 30%. That's why for severe, immediate action, it actually has to be administered intravenously to hit those peak plasma concentrations, though they have to avoid rapid infusion so they don't cause renal damage. And a huge takeaway here is that this biological battlefield isn't just a human problem. Antiviral therapies are absolutely crucial in veterinary contexts too. Drugs like encyclovir, which is a nucleoside analog that's actually even more potent than acyclovir, are vital tools for veterinarians treating serious feline and equine herpes viruses. The fundamental mechanisms of this cellular hijacking, they literally span across mammalian species. All right, our final section, part 5, halting assembly and release. So what happens if the virus manages to copy its DNA anyway? Well, we try to lock down the exits, and we have 2 distinct late-stage strategies to trap it inside the dying host cell. On one side, we have assembly inhibitors, like the water-soluble cyclic amines amantadine and rimantadine, which literally scramble the final late-stage assembly of RNA viruses like influenza A. On the other side, we have release inhibitors. Oseltamivir, for example, is an ester prodrug that competitively inhibits an enzyme called neuraminidase. Without that enzyme, the newly formed influenza viruses can't bud and release. They literally remain chemically glued to the host cell surface, completely unable to spread the infection. Oh, and interestingly, bridging back to our veterinary focus for just a second, drugs often hold these really surprising secondary secrets. Amantadine isn't just an antiviral that disrupts viral assembly. It actually doubles as a vital pain management tool for animals. It does this by targeting completely different neurological receptors, specifically inhibiting the neurotransmitter NMDA. It's a fantastic reminder of just how complex and multi-layered pharmacology really is. Before we wrap up this explainer, we absolutely have to talk about interferons. These polypeptide molecules act as the body's natural high-alert security system. Now, they possess indirect antiviral properties, meaning they don't attack the virus themselves. Instead, they induce potent antiviral mechanisms via gene promoters in the host sense, actively interfering with viral multiplication and protein synthesis. And what's truly mind-blowing is that once they induce the state in one cell, that cell can literally transfer its fortified antiviral state to neighboring cells just through physical contact without even needing more interferon. It's like a localized cascading defense network. So, as you think about all this incredible ingenuity, from fake DNA building blocks to chemically gluing viruses to cell membranes, ask yourself this. What happens when these microscopic intruders inevitably mutate to bypass our best security? We already know resistance to crucial drugs like oseltamivir can develop incredibly rapidly. So how long can our current pharmacological arsenal hold out? And what on earth will the next generation of this continuous evolutionary arms race look like?